Bold claim: an old drug for pregnancy reveals a surprising weakness in brain cancer, offering a new path for safer, smarter therapies. But here's where it gets controversial: this 70-year-old medication might do more than lower blood pressure, it could reshape how brain tumors are treated. The story below preserves the core facts while presenting them with fresh clarity and accessible explanations. Hydralazine has long been a mainstay in medicine, especially for preventing life-threatening hypertension during pregnancy. Yet for decades, scientists lacked a complete picture of how it works at the molecular level. That gap in understanding has limited insights into exactly why it's effective, how safe it is, and what diseases it could potentially target. Researchers from the University of Pennsylvania, led by Kyosuke Shishikura and postdoctoral advisor Megan Matthews, along with collaborators, have now solved this long-standing mystery. Their study, published in Science Advances, identifies hydralazine's mechanism of action and uncovers a surprising biological link between hypertensive disorders in pregnancy and brain cancer. The work suggests that established medications can harbor other, previously unrecognized therapeutic potentials, and that decoding their action can guide the design of safer, more targeted drugs for both maternal health and brain cancer. Preeclampsia has affected generations of women in my family and continues to disproportionately impact Black mothers in the United States, Matthews notes. Understanding the molecular details of hydralazine opens the door to safer, more selective treatments for pregnancy-related high blood pressure, potentially improving outcomes for those at highest risk. Key mechanism: hydralazine blocks an oxygen-sensing enzyme. The team discovered that hydralazine inhibits an oxygen-sensing enzyme named 2-aminoethanethiol dioxygenase, ADO. This enzyme acts as a molecular switch that signals blood vessels to constrict when oxygen levels fall. ADO functions like an emergency alarm that rings as soon as oxygen dips. Unlike many biological processes that require transcription and translation, ADO acts in seconds, bypassing those slower steps. By binding to and blocking ADO, hydralazine effectively silences this oxygen alarm. When ADO is silenced, its usual degraders, the regulators of G-protein signaling, RGS, proteins, accumulate. The buildup of RGS proteins tells blood vessels to stop constricting, counteracting the brain's squeeze signal. With reduced intracellular calcium, the smooth muscle cells lining blood vessel walls relax, producing vasodilation and a drop in blood pressure. From preeclampsia to brain cancer, a shared target. Before this work, researchers suspected ADO's importance in glioblastoma, a brain cancer that often thrives in low-oxygen environments. High levels of ADO and its metabolic products have been linked to more aggressive tumors, hinting that inhibiting ADO could be a powerful strategy. However, a suitable inhibitor to test this idea wasn't available. To test hydralazine's potential, Shishikura collaborated with structural biochemists at the University of Texas. They used X-ray crystallography to visualize hydralazine bound to ADO's metal center. Neuroscientists at the University of Florida then evaluated the drug's effects in brain cancer cells. Their findings show that the same ADO pathway that governs vascular contraction also helps tumor cells survive in hypoxic conditions. Unlike traditional chemotherapy, which aims to kill cells outright, hydralazine disrupts this oxygen-sensing loop and triggers cellular senescence, a dormant, non-dividing state in glioblastoma cells. This effectively halts growth without provoking additional inflammation or resistance, unlocking broader therapeutic possibilities. This research underscores how revisiting established drugs can reveal new therapeutic avenues and inform the design of safer, more precise treatments for both maternal health and brain cancer. The next steps involve refining the chemistry to craft new ADO inhibitors that are more tissue-specific and better at crossing the blood-brain barrier, thereby targeting tumor tissue while minimizing systemic effects. Matthews is also focused on advancing the next generation of medicinal solutions by elucidating the mechanics of long-known, clinically tested treatments. It's rare for an old cardiovascular drug to teach us new biology about the brain, Matthews observes. But that rarity is exactly what motivates ongoing exploration of unusual connections that could yield novel solutions. About the researchers. Megan L. Matthews is an assistant professor in the Department of Chemistry, School of Arts and Sciences, University of Pennsylvania. Kyosuke Shishikura is a Penn Arts and Sciences postdoctoral researcher in the Matthews Group. The study's co-authors represent institutions including the University of Pennsylvania, the University of Florida, the Chinese Academy of Sciences, the Georgia Institute of Technology, Nanjing Agricultural University, Oberlin College, Pennsylvania State University, Thomas Jefferson University, the University of Oxford, and Washington University in St. Louis. Funding and support came from the National Institutes of Health, the National Science Foundation, the American Cancer Society, various foundations and funds, and other university and foundation supporters. Follow-up questions for the reader. Do you see this as a compelling example of repurposing old drugs for new uses, or does it raise concerns about safety and unintended effects? How might this discovery influence how clinical trials are designed for brain cancer therapies in the future? If hydralazine or newer ADO inhibitors prove effective, should access be prioritized for high-risk patient groups first? Would you like a version focused more on the scientific mechanisms for a specialized audience, or a patient-friendly summary that emphasizes potential treatment implications and practical considerations?